Sure, we're recording here. Sorry about that. So, welcome again. I'm the secretary of the ASCCT, and the first thing I wanted to remind you of is our 5th annual meeting. So if you're not a member, you may not have heard about this meeting yet. It is going to be this September, and we're very excited because it's our 5th meeting. And also because we have a really nice program starting to shape up. We have a couple of focus topics. One is read across, and one is induced pluripotent stem cells for predictive talks. We have 2 distinguished plenary speakers, and then we will have abstracts, talks chosen from abstracts for those topic areas, but also for general communications as well. We also, during our meetings, like to have a lot of social and student-led and mentoring activities, and so we're going to be doing some of those things, and a panel discussion at the end of the second day. So I hope you'll consider joining us and submitting an abstract. The abstract's deadline is July 22nd. And you can find out the link to more information on how to submit an abstract and how to register at our website, ascctalks.org. So I'm going to go ahead and get started with the program today. First, a little bit of general housekeeping. The webinar is recorded, and ASCCT members do have access to the recordings of all of our webinars shortly after the event. Your lines are muted, but after the presentation, there will be an opportunity to ask questions. So feel free to put questions in the Q&A box as soon as they come to you. And then once we get to the question and answer session, we will go ahead and ask those questions. And you can also reach me in the chat box if you have technical problems or questions. So today's presentation is given by Dr. Xiaochen Chen and is titled Rapid 3D Bioprinting, an Enabling Technology for Creating Functional Tissue Models. Dr. Chen is a professor and vice chair in the Nanoengineering Department and professor affiliate in the Bioengineering Department at the University of California, San Diego. He's a founding co-director of the Biomaterials and Tissue Engineering Center at UCSD. Before joining UCSD, Dr. Chen had been a professor at UT Austin, and he served as the program director for the nanomanufacturing program of the National Science Foundation. Dr. Chen's primary research interests include biomaterials and 3D bioprinting, stem cell and regenerative medicine, tissue engineering, laser, and nanomanufacturing. So welcome, Dr. Chen, and thank you so much for giving this presentation, and I'll give the floor to you.
Thank you, Christy. It's a great honor and a great pleasure to be here talking to you all. So I would like to, can you see my screen?
Yes.
Oh, great.
Right.
Okay, so for today's talk, I'll give a brief introduction at the beginning, and then our motivation for doing our work. And then I will jump to the section 2 about what can 3D bioprinting do for us in the community. I will show basically 4 areas. One is creating physical geometries. We call it designer pores and shapes for scaffolds that can be used for growing cells and other functions. Then I will show that you can also create, chemically heterogeneous scaffolds by embedding, by creating the device with multiple different materials. Then I'll move on to patterning that allow printing structures with special mechanical properties. Specifically, I will talk about stiffness control, and also another parameter called a Poisson ratio. That's related to mechanical properties. And then the last part of this capability is about printing with cell encapsulation. So that really makes the name of bioprinting. You can print the cells with the gel, so the material you are creating is a live piece. So from research point of view, we use this all these different controls, mechanical, chemical, physical, and biological function to study cell interaction with the microenvironments. Then I would like to focus on a specific example that we recently worked called 3D bioprinted liver tissue models derived from human-induced pluripotent stem cells. Okay. I'll talk about how we make those and the function analysis, and also a very brief drug testing data. Okay. And then I will conclude my talk. Okay. So there are significant needs of 3D bioprint tissues. Of course, first goal is to help regenerative medicine field. Can we create device? Can we create replacement tissue for regenerative purpose? I think for this community, you are all more interested in drug discovery using this 3D bioprinted tissue. Okay. Can be animal, can be human. Okay. Especially if you use human-induced pluripotent stem cells, then you are creating patient-specific tissue models. From also in the academic field, we are also interested in biological studies using these in vitro tissue models. I want to point out that the advantage of this 3D printed tissue model is that we can create a library of changes or different models. That's hard to do with real animal or real human. Right? For printing, I can change the pore size, I can change the stiffness, I can change different cells from healthy cells to disease cells. So there's a bunch of changes associated with the 3D printed tissue. And that kind of model allows you to study disease progression. So, and there's many other applications of all these 3D printed tissue, including artificial food, biological sensing, and also biorobotics. So those are very much engineered tissue that can do multiple things. Okay. The field for creating these tissue is called tissue engineering. So usually, in order to create the kind of tissue, you need cells, and a cell cannot grow in a petri dish into a functional organ, 3D organs. So there's a need for supporting the growth of a cell, and we call scaffolds. And then, in order to have a functional tissue, you do need more than just cell and a scaffold. You need all these different functionalities to support the growth. For instance, mechanical stimulation, electrical stimulation, structure support, extracellular matrix molecules, growth factors, and even gene delivery. So with these, you could possibly create this engineered tissue for repair, for regeneration, for wound healing, and also for drug screening. Okay. From printing point of view, I'd like to mention two things. That's very related to engineering. And number one is this tissue stiffness. As you can see that in a human, all these different tissues or organs have quite different stiffness, from fat to kidney to cartilage to bones. So there's an essential question about what material we can use to create a scaffold, and that can also work with the cells. Right. So, for instance, you cannot really use ceramics material. Ceramics are very hard, tough to create as a scaffold material to work with a brain tissue. right? So for brain tissue, it's very soft. So you have to use very soft hydrogen materials in this case. The other important issues that we need to keep in mind is the dimension, because cells are on the order, in terms of size, are on the order of micron. But these cells interact with the environment at micro, even nanometer scales. So if you can, from creating tissue replacement point of view, we have to look at all these different issues like chemical composition, ligand density, network mechanics, or shapes and topologies. Right? So all these will affect the cell behavior. Okay. In terms of creating these tissue, there's different ways, and I would like to focus more on printing part. So traditionally, you see all these printers are based on either inkjet or extrusion type of machines. And these are, I have to say that these are excellent machines, and they're low cost, very easy to use. But certainly, there's also issues associated with these. And I do have such kind of printer in my own lab, but unfortunately, none of our students want to use them. There are several reasons. Number one, for inkjet-based processing, and you often get clocking, in the tubes, either the tissue, either the cell or the hydrogel or the inks. Okay. And for this protrusion-based process, you have to use mechanical forces, and that will introduce shear force, and these kind of shear force could damage the cell. Okay, and then for thermal-based inkjet printing, you also need to use heat. So certainly, if you want to print the cells with the ink, it's not easy. And on the right side, I show a cartoon that kind of typical process using either inkjet or extrusion. But basically, is a point by point, drop by drop, and line by line, and layer by layer processing. So it takes a long time to build even very simple geometry. For traditional 3D printing, that's okay. But for bioprinting, with a cell inside the ink, and that could be a problem because you don't want to keep the cells outside the culture media for a long time. For instance, if you want to print this tube in 4 hours, that's really too long. Okay. So at the very beginning, I look at this this field. I realized that we can do it quite differently from what traditional people are doing. In this case, we use the light to polymerize the photosensitive monomer solutions. Okay. Then when you shine the light, this focus spot becomes solid from liquid. So you can scan the light from one point to another, and eventually will give you the three-dimensional structures like this, this demonstration here. And another thing nice thing about light is, we have tons of supporting technology from the semiconductor industry about light control, size, and interaction with the material. So from printing resolution point of view, here we don't have clocking, we don't have shear force. And light allows you to focus down to micro scale or even nanometer scale. So on the right side, that's the work we did like 10, 12 years ago. Then we can create all these micro scale scaffolds, using such kind of light-based processing. Okay, and if you use so-called two-photon printing based on an infrared ultra-fast laser, you can create a nanometer scaffolds like this one. So these lines, in terms of width, on the order of 200 to 300 nanometers. It's very, very small. That kind of resolution is very hard to achieve by traditional inkjet printing or nozzle-based printing. But still, this is a point-by-point, polymerization process. You build a structure, and then layer by layer, it takes some time. So with my background in optics, I immediately jumped to another direction that can do everything but with much higher throughput. So on the left side, and that work was published in 2006, we use a digital micromirror array to deliver the light. So instead of having one laser beam here, we use a UV. Actually, now we can use a visible light to the monomer solution with multiple spots. And in a traditional DMD machine, you can have 2 million micromirrors within this device. So that means you can have 2 million spot control simultaneously. And you see this device, actually, you know this device. Actually, it's in every computer projector. Right? So each computer projector has this called DLP chip. It has 1 million to 2 million micromirror inside. And the reason you see the PowerPoint presentation, the whole screen show up simultaneously is because of this digital micromirror array device. So same thing, instead of sending the image from a PowerPoint to your screen, here we send the image to the photosensitive polymer. So with the light, we can polymerize it. Without light, if it's dark, then this part stays as a liquid without polymerization. So you can create this kind of device, simultaneously without scanning. We call scanningless in the XY directions. So once one layer is done, you move the stage down, and then you print another layer. And but this, in the Z direction, you have to wait until the liquid to fill up this previously solid plane. So that takes some time in terms of fluid flow and the wetting, right? So it took us a while to figure out that you can do a little bit different way when you put these things upside down. So on the right side, we actually print the material inside the liquid, because most of the hydrogel material we are working with now are transparent to near UV or visible light. Okay, so you can shine the light from the top and polymerize this layer, and then continuously going down, by moving the focal plane from the top to the bottom. Okay, so in that case, you don't need to wait until the liquid to fill the top surface. In this case, you can do so-called continuous dynamic printing. So that really helps in terms of speed. Traditionally, it takes hours to print a device. Here, it only takes seconds to finish. I'll show you a video later on. Okay. So this is a cartoon again showing traditional nozzle-based printing, and here in our case, we call scanningless printing. We don't do this point by point, then layer by layer printing. We print this whole thing simultaneously without XY scanning and without Z direction stopping. Okay, so let me show you a quick video here about this machine. I can just quick start from here, do some setting on the web or the software, then you hit print. So now you can see this guy is printing a tube out of this hydrogel solution. Okay, and you can slowly see this is coming out. So it's like a human emerging from a lake type. So everything is printed simultaneously in the XY direction, and then in the Z direction is poured continuously. So that really saved time. So this tube is about 2 centimeters in terms of length and 2 millimeters in diameters, and it's a hollow. Okay, it's hollow, so you can see how quick we can print this kind of structures. This is polyethylene glycol. You can see it's transparent. So go back to the way we print. Here, we use uncross-linked monomer. They are sensitive to light, and since it's in a solution format, you can add cells to this solution. You can add control release particles. You can add drugs. You can add growth factors into the solution, and then mix them together. Right, mix them together. Then you shine the light. So anywhere you have the light, the monomer solution will be cross-linked. Outside the light area is still as liquid. So when you cross-link these specific areas, whatever inside this area will be embedded, like nanoparticles or cells. So that's how you can create a functional devices, chemically heterogeneous or biologically active when you have cells inside. Okay. So we have been using different type of materials like polyethylene glycol, called PEG diacrylate. So you can photo cross-link them. You can tune the mechanical property by using different molecular weight, or you can use different light intensity. Basically will give you the different degrees of cross-linking density. This material is a non-toxic. Generally, it's a non-immunogenic, and it can be chemically modified, for instance, RGD, so cell can attach on it. Okay. We also work with a gelatin methacrylate. This material actually is durable in the body, and also is cell adhesive, so you really don't need to do any modification for this gelatin material if you use as a scaffold material for cell. We can also print hyaluronic acids, and this material is essentially ECM material. It's degradable as well. You do need to functionalize it to make it cell adhesives. And also, we have developed protocols that can print decellularized biomaterials from heart tissue or liver tissue. Okay. So here, when we first started this printing, actually, we just used PowerPoint software because I'm a nano engineer and mechanical engineer, so we don't have good support in terms of software. So you can do that by just using PowerPoint. So that's your slides. You have this hexagon patterns. White means light will go through. Black means there's no light. So and that will turn into a piece like this one. So white means I polymerize it. Dark means I don't polymerize, so it stays as liquid. So that's basically the patterns. And so easy to do it with a PowerPoint when you change the pattern to other shapes, and then you can print it. Okay, so it's a very straightforward. In this case, you can have multiple layer structures if you have, if you keep moving the stage. Okay, so and again, I show this cartoon here for traditional inkjet printing. You print all these dots one by one in order to form these channels. And for us, in our case, we print all these channels simultaneously without going through XY scanning. Okay, so we did this the conduits or other whatever channel we need. We can print them, print these channels simultaneously. Okay. And I will also want to emphasize that really it doesn't matter how complex the structure is, you can easily print them in a matter of seconds. Okay, so here all these patterns, pyramid, spiral, dome, flowers, they can print simultaneously. You don't need, if you don't want a uniform pattern like this one, you can create gradient of the structure or topology, different features. Different distance, so it's very easy. You print the whole thing simultaneously. And since it's a continuous printing in the Z direction, I want to show you that here you don't see the layers between in the 3D device. You can see very smooth structure because you print this one in a continuous fashion. There's really no stop between different layers, and you often see those kind of layers when you do it in traditional printing. If you move from one layer to another, you have these artificial interfaces. And these interfaces between the drops and the layers often cause structural integrity issues, especially if they are really not a well-fused parts. So if you do pulling test, these artificial interfaces will tend to fail first. But with continuous printing, you can create this very integrated part without seeing these artificial interfaces. Okay, so on the right side you can see that we print this kind of vasculature network type of pattern, and these pattern can be very hard to generate using traditional printing because number one, these vessels here, or the smaller ones are small as 10 microns. And that's very hard to do with traditional inkjet printing or nozzle-based print because you make these patterns by fuse all these droplets together, right? So if you have a mechanical engineering background, you know how small droplets you can deliver, they can see immediately the limitation of them. And the second is if you are printing structures like hyaluronic acids, these are based on hyaluronic acids. These materials are so soft, it's to hold them together. If you do it point by point, then layer by layer. But if you do it all way using this continuous printing, you print the whole thing simultaneously, then they support each other. You can form structure out of these extremely soft materials. Okay. So I call this 3,000 times faster because really you don't have XY scanning. You also don't need to stop for the Z directions. And this whole thing can be printed in a matter of seconds instead of hours. And this speed, again, is very important if you are encapsulating cells in the gel, right? Because you don't want this cell to be outside the culture medium for too long. And this fractal pattern, I guess, is just for fun for us to create these engineered structures. Okay. So let me move on to second part of this printing capability called printing heterogeneous scaffolds. Means you can add different chemical functions to the device. Right? You don't need to have a scaffolds out of same materials, to be functional. So as I mentioned that you print the structures from liquid to solid. So if I print this red part first, wash out and put a green part here, then I can have a chemical heterogeneity in this device. So for this, in this case, it's the same layer, and in this case, it's more different layers. I can change the materials from one layer to another, right? I can create grading of these kind of devices by controlling, how the material delivered to the pool before you print. Okay. So another way to have different chemical control is to add, other materials like nanoparticles. So in this case, we print the structures that's embedded with polydisaccharic nanoparticles. Okay, and these nanoparticles actually can sense and trap molecules, toxic molecules in the solution. Okay, but anyway, we print them in a gel. So this is in a gel is like a sponge. It has these nanoparticles inside the gel because it's embedded when you cross-link them. Okay, and we did a simple test for this polyethylene glycol-based hydrogel device. In this case, there's no nanoparticles. Then you immerse this device into toxic molecule containing solution, and nothing changed over time. But if I put nanoparticle in the gel, then these nanoparticles can trap these toxic molecules into the gel. It's like a sponge. And then, this actually, when they trap it, there's a bonding between the toxic molecule and the nanoparticles. That bonding gives you actually a fluorescent signal. In this case, it's red. So you can see gradually these toxic molecules are diffused and then eventually trapped by this hydrogel device. Okay, so this actually is a cartoon showing you have the nanoparticle embedded inside the gel, and once you immerse this hydrogel device into a toxin containing solutions, then you can remove the toxin and by these nanoparticles. So when you take away this overall device, then you're basically doing this so-called detoxification process. Okay, and in terms of detoxification, we can think about liver. So liver actually has this so-called hexagon pattern in terms of microarchitecture, and we, from printing point of view, we can easily create these hexagon patterns. And we embed a nanoparticle inside, and we can create multiple levels of the device. So this is SEM picture. This is the fluorescent particle pictures. Once this device actually traps all these toxic molecules, in this case specifically is a melatonin, right? Melatonin. And we show that you can remove these toxins in terms of efficiency by 100%. And we studied this behavior and the function of the device by comparing it with different controls and different scenarios of the device. Okay. I'm not sure how many of you have watched this old movie called Fantastic Voyage. It's a science fiction movie about a crew of scientists, medical doctors, engineers aboard a submarine. And then this submarine is shrunk into 1 micron and it got injected into the patient who actually is suffering from a life-threatening blood clot in the brain. Okay, so I like science fiction, and when I watched this movie, I realized that hey, we can do something in terms of creating such kind of nanobots or microbots. Okay, so that actually triggered us to do this work called 3D printed artificial microfish. Okay, so for moving in bloodstream, we need to think about something in the nature that can move in the solution, in water, for instance, and fish is one of them. That's why we pick fish. And from 3D printing point of view, it doesn't matter what shape you need. So fish or other stuff we can always print. Okay, so it's a hydrogel fish. So in order to move the fish in the solution, we can put a platinum nanoparticles, and these nanoparticles can react with the solution, and that reaction can generate a bubble. Okay, when you have bubble generation, and then due to momentum balance, the fish has to go forward. Okay, but in this case, with only nanoparticle, platinum nanoparticle in the tail, you can see the fish just run circles like we call drunken fish. So how can we make this fish more like a controllable, like a navigation purpose? So we put a magnetic nanoparticles in the head. So you can use magnetic field to guide the fish by the magnet outside. So in that case, you can see this fish is no longer drunken. You can control them in terms of direction of the moving. Okay. And again, from printing point of view, we can print a school fish instead of doing 1 by 1, right? And we can change the size of the fish if you want. Just change the palm point design. And actually now since we have more, we have been working on this for the last 10 years, so we have more support in terms of software development, and we don't rely on PowerPoint software anymore. We have much better software by taking any digital data you have, such as CT scan data, MR data, and we can use digital data into a device. So from clinical application point of view, that's actually very significant. Okay. And this figure shows that the fish has different composition in terms of chemical materials. So here in the tail you have the platinum nanoparticles. In the head you have magnetic nanoparticles. Okay. And actually in the fish body we put this polydisaccharin nanoparticles inside, so the fish actually can remove toxins in the solution. Okay, when they move around. Okay, so we this is basically for us it's fun to work, but for some reason the general public is so interesting that we've got press reported by the Wall Street Journal, Washington Post, and Forbes. I think in the long run, you can see these nanorobots that can self-propelled. They can do sensing. They can deliver drugs if you want them to do, and they can even do these nanosurgery actions if you can make this fish more functionalized. Okay, so there's a really lots of possibility in the blue sky. Next, I'd like to move to this so-called patterning mechanical properties. As I mentioned that. The device you created can be functionalized in addition to chemical to physical. You can also put a different mechanical functions. And in terms of mechanical functions, first is stiffness. As I mentioned in my first couple of slides, stiffness is very important. cells are very sensitive to stiffness, especially stem cells. And you can control the stiffness by either changing the molecular weight of the materials by changing the solution, or you can control the light, how much light you put for cross-linking. So in this case, we change the light. You can see this line is stiffer than the surrounding gel. Chemically, they're the same, but physically, these lines are more stiffer than the surrounding areas. And FM, we use atomic force microscope to measure stiffness. These lines are 10 times stiffer than the surrounding gel. Okay, and physically, topology-wise, you don't see that. Okay, and then we do some very simple cell studies. We put a smooth on top of this gel. We culture them. In day one, they have very much uniform deposition. Then after a couple of days, day 4, day 6, we start to see these cells migrate to stiff areas. Okay, and this is a fluorescence image to show that these cells very much like the stiff lines instead of the soft gels. Okay, soft gel. And this picture actually was displayed in the San Diego International Airport back in 2014. Okay. Another mechanical property is called a Poisson ratio. You can do a quick experiment when you stretch a rubber band or other soft materials. You can see that you can make it longer, but then in the z direction, in the y direction, this material becomes smaller and smaller, and eventually will break. Right. So I would say 99.9% of the material behaves like that. We call it positive Poisson ratio. When you stretch them, they become smaller. But from 3D printing point of view, we can create a material that is different from traditional material. Here, when you stretch them in the y directions, the x direction, actually, the material expands. Okay, and when you release the stretch, they can go back to original state. So that's basically called a negative Poisson ratio material, because when you stretch it, instead of shrinking, it becomes larger. Okay. So the reason you can have this kind of negative Poisson ratio property is because of the microarchitecture of the material. So if you have like pore shaped like this one or this one, then when you stretch them, they actually expand. For traditional material like square holes or circular holes, when you stretch them in this direction, they can become smaller, and eventually break. Okay, so this is positive. These two devices are negative in terms of Poisson ratio. Our students love to have fun, and here they create so-called hybrid material. When you stretch it, one side it shrinks, on the other side it expands. Okay, so you don't see these materials in the nature, but certainly as an engineer, we can always create these different versions of the materials. You can have so-called zero Poisson ratio material, means when you stretch them, they don't change in the wide width in this case. Okay, and we hope someone can find uses of these novel materials. And last, I would like to talk about printing with cell encapsulation, because that's really this community about, right? You want live tissue models to play with. So as I mentioned that in the printing, you can add a cell in the monomer solution and then cross-link the gel. So when we polymerize them, then these cells will be encapsulated inside the gel. So the best way to show the cell can survive this kind of process is to use the cardiac cells. For cardiac cells, if you put them in the culture, day 5, day 7, they start to beat. So here, this hydrogel is not a regular hydrogel. It's a live hydrogel with cell embedded in that, and especially this cardiac patch, the cell can beat, and when cell beats, the whole hydrogel is moving around. I mean, is vibrating. Okay, so in this case, we just uniformly put cells there. You can see the beat, but if you do a little bit engineering, by creating the pattern, or lining up these cells, you can see immediate difference between this case and the other case. So in this case, these cells are better organized. They have synergy. They talk to each other better, and they beat differently. Okay, and this so-called optical mapping show the difference in terms of the magnitude and the frequency of the beating. Okay, of the beating. Okay, um, to show some really useful model, I would like to use this very recent work we did as a example here. I've got 3D bioprinting a human liver model. Liver. If you look at the liver in terms of microarchitecture, they have this hexagon pattern for these hepatic cells, liver lobule. And then you have central vein. You have also this connective tissue septum here. So that's basic architecture. If you want to create a human liver, I think we need to. It will be great if you can create such kind of biomimetic architectures. So to do that, you need to have complex architecture. You cannot just put a cell with the gel and hope that it's a great liver. Okay. Number two. This tissue model has multiple cell types, right? So we need to create cow culture or even trial culture models with the liver cells and also the supporting cells. And the last thing, you need to have these extracellular matrix cues. So what kind of material you're going to use will affect the function of the liver. Okay. So we developed this so-called two-step printing process to create this human liver model. For step one, we print liver cells with the gelatin materials. The reason we use the gelatin material is because the stiffness is very close to the real liver of the hydrogel. Okay. As I mentioned, that I mix the cells with hydrogel, and then I shoot the hexagon pattern to the gel, okay, and in a matter of seconds, we can print this piece of hydrogel with liver cell encapsulated. In this case, we use a human-induced pluripotent stem cell-derived hepatic cells. So it's a human liver tissue here, and this is for liver part. So I intentionally leave behind this open space between these hexagon patterns. We try to put HUVEC cells in these areas, in the dark areas. Okay, so you wash out these whole things, and then you have an open space to create HUVEC cells for in these areas, in these open areas. So here we use gelatin with hyaluronic acids, because acids can help cell growth, vasculature formation. That's why we mix the gelatin with the hyaluronic acids. And we also added another supporting cells, adipose-derived stem cells with HUVECs for the cell part. So again, this mixture is deposited to the solution, to the piece that we previously printed. They can fill up those open areas, for the central vein part, also for the hexagon edges. So in another second, we print the HUVEC cells, ADC cells in these areas along the side of the liver, lobule. Okay. So if you look at the merged pictures here, you can see this very nice, precisely defined human liver tissue model. Okay, so with hepatic cells in green and HUVEC cells and adipose-derived stem cells in red in these areas. Okay, and this is actually 3D reconstruction of the first microscope picture. All right, so in the day one, you can see these cells are there in terms of culturing, and over the when you have more time to culture them, you can see these cells start to integrate with each other. But even day 7 can still see very clear pattern of the HUVEC cells in this tissue model. Okay, so and it's very important for us to study whether these are the liver or not, right? So So we do gene expression studies here for look at specifically 4 key liver genes: albumin, TTR, and HNF4 alpha. Like you can see, this is our model by 3D printing. We call 3D triculture model because it has liver cells, it has adipose-derived stem cell, it also has a HUVEC cells. So we call triculture model. For the study itself, we try to compare with other cases, the 3D, 2D mononuclear case, and also 3D hepatic cell only case. Right. So in this case, even it's 3D, but we don't put supporting cells in the pattern. Okay. So you can see there's a significant difference in terms of these liver specific gene expressions. The 3D triculture model has a much higher level in terms of albumin, HN4 alpha, and TTR. We do see this expression for the AFP here, fetal hepatic marker, not so significant, but you can still see the reduction of this specific gene. Okay. And next, we want to look at anabolic and catabolic liver functions. So specifically in the albumin secretion level and urea productions. You can see this 3D triculture model has much higher such kind of levels in terms of albumin and urine in comparison to 2D and also 3D hepatic cell only models. Okay, so it looks like this liver, 3D printed in vitro liver is functioning. Although we do see the reduction after this day 10, this actually is the number of days following bioprinting. So after day 10, you can start seeing this decrease. I think the reason behind this one is this iPSC-derived hepatic cell is really not mature enough in comparison to real human liver cells. So I think there's a work to be done to make this iPSC-derived liver tissue more mature. But definitely, it's the data support that this is a useful model in terms of function and gene expressions. So we quickly jump to something wrong with my screen here. Jump to this very simple testing because the best thing out this in vitro model is to it's also iPSC derived model is to do drug testing. So we just pick one, that's pissing, we say antibiotics, and you can see this treatment can lead to significant increase in these 4 enzymes: CYP3A4, CYP2C9, and 2C19. Okay, for our specific triculture models, and the other 2, 2D monolayer or hepatic cell only 3D model doesn't show significant difference between the control and drug response. So I think this pilot study actually show it could be very useful for drug screening. Okay. We are working on with some other groups and companies try to test unknown drugs because in this case it's known drug. So hopefully this will get more data for that. So to summarize my talk, I have shown you that this light-based processing can get you better resolution in terms of printing, and more importantly, can get you much faster printing speed. Okay, and that printing allows you to create functional scaffolds with specific designer shapes, with unique mechanical property like stiffness and Poisson ratio. You can have good control in terms of chemical composition, and also cell encapsulation. Not just one cell, and also multiple cells. You can put them in a very well designed location to study either drug response or disease modeling. So we have also demonstrate the use of various biopolymers such as polyethylene glycol, gelatin, hyaluronic acids, and their combination as for precision tissue models. So I like to thank my students over the years. Many of them actually have working in industry. Some of them are also finding academic jobs, both in high tech. I mean, academic jobs, in different schools, and we usually maintain good a sizable group with PhD students and also postdoc researchers. And I appreciate very much all these collaborators over the years here at the UCSD and the Harvard Medical School. And the group has been funded by National Science Foundation, National Institute of Health, and also we have a grant from California Institute for Regenerative Medicine. So with that, I'd like to conclude my talk. And if you happen to be here in San Diego, you're welcome to visit us. Thank you very much.
Thank you very much, Dr. Chen. That was a very interesting talk. And a really nice introduction to some of the issues to consider for bioprinting and for cellular models. You mentioned, well, let me invite others to to ask questions. Again, if you want to ask a question, please type it into the question box and I'll be able to ask it for you. And I have a quick question while we see if others are going to ask one. So you mentioned some collaborations with some other groups. I'm curious if you've been talking with, if there's interest with the pharmaceutical industry or the FDA, and if you can kind of share what you're looking ahead to do in that realm.
Sure, yeah. So we have, being here in San Diego, we have so many biotech companies around us. And I've been talking to many of them, Pfizer, Johnson, and Takeda, and Novartis. And actually, some of my students working in Novartis, so they try to work with us in terms of creating some of the tissue models. I haven't done any talk yet with FDA, but they came here one time to learn what we are doing. So I think next step probably is try to open up this dialogue channel with FDA, see how we can take this to a commercial levels. We do have patents, provisional patents for many of these work projects. Ideas here from the lab.
Okay, great. Yeah, I think that's one thing I've learned over the years. And actually, one of the reasons our society is set up the way it is to try to really promote that dialogue between regulatory agencies, researchers, and industry, because the earlier the better, we have found when trying to create new ways of assessing biology or toxicology. So
I agree. Yeah, the earlier the better. But from research point of view, we wanna, we do not want to confine our minds. We want to really check out all these possibilities. So I think definitely in the future we should walk with the two legs.
Yep, that's right. Okay, well, I don't see any additional questions coming through, so it seems like you had a very clear and interesting talk, and so I'll thank everybody for attending. You will receive, ASCCT members will receive a link to a recording of the webinar. And if you would like more information, please just email me or visit our website. Oh, we have a very nice thank you. Thank you for a nice talk. So that's great.
Thank you all.
All right. Well, thanks very much, and everyone have a great weekend.
Thank you. Bye.
Bye.